Hello, this is me, Rim Aporipan, and I will talk about the amino acid. So I will first start with the most, or we can say it's the simplest amino acid, which is known as the glycine. As you can see in the photo, we have the glycine, which is the simplest amino acid that is composed of the alpha carboxylic group, and we have also the N-terminal, which is the amino group, and the carboxylic group, which every amino acid has it. So we have the carboxylic group, we have the amino group, and the carbon alpha, which is the central carbon, but there is no R group because it's HH. So that's why it's the simplest amino acid. So its initial, that's mean the first letter of it is G, and that is depend on the pronounce of the glycine. So we can say glycine, so the initial is G, and the nickname is Gly, Gly. So again, it's the simplest amino acid. And the other thing is optically inactive. Inactive, that's mean we can see it's inactive, but when it's combined with the other compounds, then it become active. But it's optically it's inactive. Non-essential amino acid, which is mean that we don't need to take it in the diet. That's mean we don't have to eat it in the diet. The body itself form it, which is 3 grams per day, and also you can eat it. Maybe you can eat it present in the food, which is about 3 grams per day. So completely is 6 grams per day. But again, non-essential amino acid. That's mean you don't have to take it in the diet, you know. And so I will talk about a point also about the glycine. When we combine it with the CH3, then it's formed something which is known as unmethyl glycine, and when it's formed unmethyl glycine, then which is known as a sarcosine. So when you hear a sarcosine, now you know that is unmethyl glycine. So we add the CH group methyl group to the glycine, and that is formed something which is known as a sarcosine. And also the amino acid, which is known as a serine, can convert to the glycine. And if you look the photo here, so we have the serine by the addition of the vitamin 9, B9 and B6, and that is will remove the B carbon or the beta carbon, and that will lead to something which is known as a glycine. So we can turn from the serine to the glycine by the removal of the beta carbon and by the help of the B6 and B9, and that it will lead to the form of glycine. Uh, Now we're talking about the functions of the glycine. The first one is creatinine synthesis, and we know that creatinine is the waste product of the kidney. So the creatinine is formed of 3 amino acids: glycine, arginine, methionine. I don't care about the other amino acid. I'm just focused now on the glycine. So the creatinine is formed of glycine. Glutathione synthesis. The glutathione synthesis also is formed of glycine, which is formed of glycine, cysteine, and glutamate. So it's formed also of glycine, and glutathione act as antioxidant. We have also that the glycine is so important for the heme synthesis, and we know that the heme synthesis, which is lead to the formation of hemoglobin because it composed of heme and globin. So heme is formed of glycine and succinyl coenzyme A. So it's the CoA. So it forms of glycine and succinyl CoA. So it's important for the heme synthesis. It's important also for purine synthesis, and a purine, which is a part of the DNA and the RNA, and the purine is formed of glycine, aspartate, and glutamate. So it's forms of glycine. Hepatic acid synthesis, which is formed of glycine and benzoyl CoA. So it forms also of glycine. So we can recognize that the glycine is so important for the synthesis of many important things in our body, like creatinine, glutathione, heme, purine, and hepatic acid. So we can say that the glycine is an important, and I just want you to remember the glycine has no R group, or the R group is H, we can say that. So it composed of a C carbon carboxylic group and terminal or amino group and 2H. So we can say that the glycine is so important for the conjugation of the bile acid. So we conjugate the bile acid with the glycine or a turine to form something which is known as a bile salts. So the bile salts is so important for the fat emulsifications, and I mean by that it increases the surface area of the fat so it easily to be absorbed in. So which glycine it's an important things for the conjugation of the bile acid to turn it into the bile salts. Okay, I will now mention some facts about the glycine. The first one is the molecular weight is 75 grams per mole, and that is indicate that it's the simplest amino acid. It's also the smallest amino acid, and it's so flexible because side chain is only H group, so it's so flexible. It's so small, this can easily move, so it's so flexible. And the other thing, the glycine by the glycine cleavage complex, and that is lead to the turn of the and NAD positive to the NAD H and H positive, and that is will turn to something which is not carbon dioxide and ammonia. And I think that is an important reaction that we have to keep it in our mind, because maybe that will we will need it in the future. So keep in mind that the glycine by the glycine cleavage complex and by the NAD positive to turn it into the NAD H and H positive, and that will turn to the carbon dioxide and ammonia. It acts as amphoteric. That means it can be act as acid and the base, and that is because of the H group, So it can as amphoteric acid and base, and that why it used the glycine as a buffer in the lab, because the buffer we needed is a weak acid and a base, and it acts like that. It's the most abundant amino acid in collagen, and it's colorless, sweet tasting, and crystalline soil. It colors, it has no color, sweet tasting, and that way it's will discovered by the scientist, and you just taste it and you find it is sweet, and that's why it's called glycine. And it's a crystalline soil, and due to the one hydrogen side chain, it can maybe hydrophilic or hydrophobic. That's mean water loving or water hating, as I can say. So the glycine is also a neurotransmitter inhibitor. It causes interference with the release of the neurotransmitter signals within the spinal cord. So glycine is a neurotransmitter inhibitor, and that is will lead to inhibit of neurotransmitters signals. Also, glycine is used in the synthesis of the SDS page, which a method of protein analysis, and I will discuss it later. It's important. This I think it's so important for the protein analysis, and the glycine is a one component of the SDS page. It acts as a buffering agent which maintains pH. Again, I said the glycine, it acts as a buffer agent and maintains the pH and to prevent the sample damage during the electrophoresis. Glycine also is used to remove protein labeling antibodies in the Western blot. So now we understand many informations about the glycine. I will keep the PDF in the comments so you can print it and get mm, uh, benefit from it. So please, if you like it, subscribe, like it, and share it. Thank you.